Can anybody tell me like what portion of the digestive tract or what organ of the digestive tract we're looking at here? The small intestine, yeah. I love all Okay.
Yeah.
So we've got several different structures of the small intestine which make it a perfect organ for absorption. That's where the bulk of nutrient absorption is actually going to occur. One of those is going to be circular folds, one of them is going to be villi, and then finally you'll have microvilli. Looking at this structure, you've got these sort of finger-like projections. What are these of those three options?
Microvilli.
Villi. So does anybody remember the type of epithelial tissue you find lining your small intestine, stomach, as well as large intestine? Columnar. Yeah, simple columnar. And you'll notice here, like, those are the individual columnar cells. And then you can see those blue guys, those are the goblet cells secreting mucus. The microvilli, you would not be able to see in this model. They'd be kind of projecting off of these individual cells. Thus, these things, those are the villi, not microvilli. So your mucosal layer basically consists of three structures. You've got the epithelial tissue, you've got the lamina propria, which is going to be just connective tissue, like loose connective tissue fibers, which will also have like the blood vessels. And then finally you have this layer right here. So from here forward, that's all the mucosal layer. What is this layer right here? Muscularis mucosa. So mucosal layer. What then is this layer right here? Submucosa. Submucosa. This forward is the mucosa. Let's see if I can get it this way. Then you've got two layers of muscles here. What are these collectively called? Muscularis externa, and then finally you have the serosa. Now, if we were looking at the esophagus, what would it be called? The adventitia. And it basically just depends on like the type of tissue you find, but it's a little different for esophagus. Now, you've got a lymphatic structure which helps you absorb lipids. It's this green structure right here. What is this called? A lacteal. This green structure right here is called a lacteal, which helps you absorb dietary fats. Now this model's a little bit misleading because we talked about a couple different structures found only in your duodenum versus your jejunum and ileum. This model is showing you both kind of those structures that you find in the duodenum and the ileum. One is this big guy right here. Does anybody know what this is called? The Peyer's patch. What about this right here? Two names for it. This would be the one in your duodenum. This would be found in your ileum. Yeah, B. Close. So it's called the Brunner's glands. Brunner's glands or the submucosal glands, and then this is a Peyer's patch or an aggregated lymphoid nodule. So you could see this structure on your exam. It'll look pretty similar to the actual histology you'll see under the microscope. All right, who is this?
Liver.
This is the liver. How many lobes does the liver have that we care about?
4.
4. What is this one right here?
It's a
Quadrant. And that's one right next to this guy right here, which is the gallbladder. What does the gallbladder do?
Stores bile.
Yeah, it stores bile. It stores and concentrates your bile. You have a duct going directly into the gallbladder. What is this duct right here? The cystic duct. What is this lobe over here?
Right.
This is the left lobe. Now, there's a duct here. What is this duct called? Yeah, the left hepatic duct. Now, what's this big lobe out here?
Right.
The right lobe. And what is this lobe, or this duct?
Right.
Right hepatic duct. So then those two merge and become the common hepatic duct. Common hepatic duct and the cystic duct merge to become the bile duct. What is this lobe right here? Caudate lobe. What is this ligament right here? Falciform ligament. Very good. What's the cell of the liver that makes bile? Hepatocytes. Which brings me to this model right here. So this is a model of liver histology. What are these hexagonal structures right here? These are the lobules. Remember, a lobule is always going to be much smaller than a lobe. Same with like ductules versus ducts. At the dead center of a lobule, you have the central vein. Yeah, central vein. Then you've got these three structures right here. What do we name them collectively? The portal triad. And what are they named individually? For instance, what's this? Hepatic artery, hepatic vein, or hepatic portal vein, and then yep. Here on this side, you can actually see like the individual hepatocytes. What else did I mention? And then over here again, that is your portal triad. All right, what is this? The bronchial tree, exactly. Up here, what is this? Your voice box. The larynx. What is this cartilage called? Thyroid cartilage. And here, cricoid cartilage. Just remember, your thyroid cartilage is not continuous posteriorly. Now, from about here to here, what is that? The trachea. You've got cartilaginous rings, again, that help keep that airway open. What is the point at which the trachea splits?
Carina.
The carina, good. You've got this and this. What is this structure? Primary bronchi, is it right or left? The right one, yeah, this is the right one. What about this one?
Left.
The left one. So primary bronchi or main bronchi. Then those are going to branch into the secondary bronchi or lobar bronchi.
Yep.
And then these individual ones are the
Tertiary
bronchi or segmental bronchi. So here we have two. Now this one is a little bit harder to see the third. I don't know if it includes it. But for instance, how many lobes does your right lung have? 3. What are they called?
superior.
Yeah, superior, middle, and inferior lobes. And then you've got 2 fissures. Does anybody have lungs in front of them? You guys have some. Got that. Thank you. Which lung is this?
Right.
The right lung. What's the name of this lobe?
Superior.
What is this fissure?
Horizontal.
Good. This lobe?
Middle.
Middle. What is this fissure here?
Oblique.
And then this lobe?
Inferior.
Lungs are easy. Left lung, what is this lobe?
Oblique.
Honestly, that's like the gist of the lungs, but definitely know the histology as well. Alright, who is this?
Stomach.
The stomach. What is this structure right here? The esophagus. Now you kind of have this sort of rounded region right here. What region of the stomach is that?
Fundus.
The fundus. Anytime you see like kind of like rounded region, for instance, we have it with the bladder as well.
It's like
So that would be the fundus. Same with the stomach, fundus. The uterus has another one as well. So I don't think there's anything else solid out in front of me. Not histology, first model. Now looking at the stomach, what's unique about the stomach's histology as far as the muscularis externa? How many layers does it have? The 3, yeah. It's gonna have that same circular and longitudinal layer, but it'll also have an additional oblique layer. Now looking at the stomach, you've got these sort of invaginations right here. What are those called?
Rugae.
Rugae. So when the stomach fills up, are those gonna smooth out, or will you see those? Yeah, those will smooth out. Now this region that kind of connects immediately to the esophagus, and what is the name of the sphincter that separates the lower esophagus in the stomach? Lower esophageal sphincter. What type of muscle will you have?
Smooth.
In your upper esophageal sphincter, it'll consist of skeletal muscle. So what is that region called right about here?
Cardiac.
The cardiac. Right? Now the bulk of the stomach is the
Body.
body, and then what is this region here? Pyloric. What is the sphincter that separates the small intestine and the stomach?
Pyloric sphincter.
Pyloric sphincter. So then what is this first section? Duodenum. Easy peasy. Alright, who are these?
Kidneys.
Kidneys. What is this muscle right here? Yeah, the abdominal aorta. What about here?
Inferior vena cava.
What is this vessel?
Renal.
Renal arteries. What about here?
Renal veins.
Renal veins. Now, so this is your kidneys. What are these right here?
Ureters.
Your ureter. So those are going to take the urine made in the kidneys and bring it to the
Bladder.
bladder. There. Actually. This we're looking at an anterior view because you can see that pubic symphysis right here. I'm just going to remove this for ease of looking at it. You have this area of clinical significance marked by the 2 openings for the ureters here, and then the opening to the urethra. What is that called? The trigon. You're going to have rugae as well in your bladder, and then what's the muscle in your bladder? So that's going to be the epithelial tissue that's basically close, like here. This is all going to be that uroepithelia or transitional epithelium, but you also have a specific muscle. Basically, it's 3 layers of smooth muscle. What is that muscle called? It starts with a D. 2nd letter is E. So it's called the detrusor muscle. The detrusor muscle. And then finally we'll head into the urethra. got two sphincters. One will be the sphincter, your internal urethral sphincter. What type of muscle do you find that regulates the internal urethral sphincter? Smooth muscle. What about the external urethral sphincter?
Skeletal.
Skeletal. You guys are gonna crush this in the end.
I hope so. At least it's multiple choice. I have a kidney on my screen now. The giant kidney taking up half my screen.
Okay, is this female or male anatomy?
Female.
Female. What is this structure right here? The urethra. So this is the
Bladder.
the bladder. So what is that muscle again that makes up the bladder? Detrusor muscle. So that's going to be the more anterior. What is this structure then? Uterus. Good. What are the layers of the uterus?
Parametrium, myometrium, endometrium.
Yeah, good. The parametrium, the myometrium, and the endometrium. Which of the layers is shed of the endometrium each cycle? So remember, we've got two layers. What are those two layers? What's the layer that's like continually regenerating? Think back to when we did like the layers of the skin, or the strata of the epidermis. It was that lowest layer. It's similar. I assume it just has to do with being a stem cell.
Stratum of functionality.
So that's going to be the layer that's that's shed each time. What's going to be that layer that continually regenerates it?
Basal layer.
The stratum basale, or stratum basal, or basal layer. What is this structure here?
Uterine tube.
The fallopian or the uterine tubes, and that's going to be where fertilization actually takes place. What are these kind of feather-like structures right here? That like fimbriae, yeah, that basically sweep the egg or the ova once it's released. So then that makes this whole thing here.
Ovary.
The ovary, yeah, good. What is this structure right here? The vaginal canal, and then right here, this would be the cervix. I in the past have not asked about ligaments. I don't know if I'm gonna start today on Wednesday. I mean, what does anybody remember what the structure would be that connects the ovaries basically to the uterus?
Is it no.
The ovarian ligament, yeah. Sorry, Alicia, I knocked your phone over. Also, Alicia, it says your storage is full. Alicia. Again, if you guys
Are you
have
deleting
watched this
pictures?
YouTube video that's posted, they will have pretty much all of this. They might go a little further into detail than I do, but
A couple months. It's crazy.
okay. Is this male or female anatomy? This is male anatomy.
They're crazy.
What are the male gonads?
Testes.
What are the male gametes?
Spermatozoa.
Yeah, spermatozoa. What about for females? What are the female gonads? The
Ovaries.
ovaries. And what are the female gametes? The ova. Okay.
I didn't realize I just got totally angry about the picture.
So this is going to be the male urethra. What is this structure here called? The spermatocord. What is this thing right here where sperm will go through maturation and capacitation?
Epidermis.
Epidermis, good. And then what is this? The the testes. Okay, what is this structure right here?
Bladder.
It's just the bladder, yeah. What is this purple structure right here?
Prostate gland.
The prostate gland, good. Nothing much else I've asked. There is another model where you can see more of the seminiferous tubules. Oh wait, let's see here. That might be easier to tell. That's the bladder here.
Yeah.
We definitely need to do the heart. Remember, you will see a dissected heart on this image.
I need a nose plug.
Remember how I said what?
I need a nose plug.
Is that the stink?
Yeah.
I told you that the brain was my least favorite thing. Or like the school.
Mm.
I lied, it's the heart.
Really?
Yeah.
I kind of like the heart.
I just think I don't give a shit. It's
Yeah.
just the problem.
Yeah, I'm like done this semester.
Like, I never
Okay.
have, like, actually fully remember anything from the heart because I definitely don't care.
What is this outer
Like, probably
layer
never did find
surrounding
it.
circulation?
Mm,
The capsule.
capsule.
What about this lighter tissue right here? Includes the portions that extend here.
Cortex.
The cortex, and then what are these?
Medulla.
The renal pyramids, which are made of are considered the renal medulla. Good. So, what are the tips of the pyramids called? The renal papilla. Yeah, exactly. The renal papilla. And then you've got after filtrate is created within the renal. Say again? Oh yeah, minor calyx, exactly. So minor calyx, after your filtrate is collected into the collecting duct, we can see the one right here, and we'll zoom in on it shortly. Basically, that filtrate will be taken into the minor calyx, and then into the
Major.
major calyx, and then into the
Or the renal.
Trunk.
yeah, the renal pelvis, you know it, and then the ureters. Blood supply to the kidneys. What is this? Renal artery, and then into the what do you say?
Systemic.
Not systemic, but it does start with an S. Segmental, good. Segmental arteries, and then into the interlobar arteries, and then into the arcuate arteries, and then into the. Did you say cortical radiate?
I think corticate.
You know, it's close enough for me. Cortical radiate, and then you might for lecture see the term interlobular. And again, the same thing. We know lobules are going to be smaller than lobes. So this is the interlobar artery. This is the interlobular artery, also known as cortical radiate, because basically they're radiating outwards into the cortex. Now to really see what vessel comes next, I'm going to bring you over here. So pretend that your cortical radiate artery is bringing blood to this vessel. What is this vessel? Afferent arterial. What about this? Glomerulus, also known as the glomerular capillaries. Does anybody know what renal exchange process actually takes place at the glomerulus? You've got filtration, reabsorption, secretion, and excretion. It's a lot of physiology loaded question, but at the glomerulus you only have filtration occurring. So basically you're filtering the blood outward, and then that's going to enter this structure here, which is the PCT. But I'm going to finish the blood supply before we get into the tubules. So glomerulus, you've got filtration occurring, and then you head back into the efferent arterial grid. Now, depending on the type of nephron, will determine what capillary it hits next. For the most part, the majority, it's going to be these capillaries up here, which are known as the, so vasa recta are the ones in the medulla. What's the other one? Starts with a P. Paratubular, very good. Paratubular capillaries. So these are the paratubular capillaries, these are vasa recta. I don't know if many T's really differentiate between the two, but just in case you do have to take your exam with another class, these ones down here are the vasa recta, these are the paratubulars. So it can be either or after you leave the efferent arterial. And it's within these capillaries that you have the other renal exchange processes taking place. That's going to be reabsorption and secretion. Okay, now we're gonna head out. What is this vessel right here? Yeah, the cortical radiate vein. So again, we're basically just gonna backtrack and exchange artery for vein, except with one exception. Cortical radiate vein here, then what's this thing right here? The arcuate vein. Let me go back over this way. So again, the blue vessel is the cortical radiate vein, arcuate vein. Now what's next? Interlobar vein, and then for whatever reason we don't have segmental veins the way we had segmental arteries, so finally after that we're going to head into the renal vein. And renal vein returns blood to the inferior vena cava. Here's our nephron. Again, you can see that cortex here, the renal pyramid here, which is the medulla. The renal corpuscle portion of the nephron is made up of two structures. You've got this kind of outer structure here. What is that, like, outer structure of the renal corpuscle? The Bowman's capsule, and then again, what is that set of capillaries right here? Glomerulus. So this structure that branches immediately off of the Bowman's capsule is the PCT, that is the proximal convoluted tubule, and then you head into the loop of Henle, and then into the distal convoluted tubule. And that marks the end of the nephron because this structure is not part of a single nephron. It collects the filtrate made by multiple nephrons. What is this thing again? The collecting duct. And over here, this is basically just the Bowman's capsule out here, the nephron, the glomerulus, and your afferent arterial, you've got a portion of the juxtaglomerular apparatus, just called the juxtaglomerular cells, and those are the cells that secrete something called renin. It's an enzyme, part of this renin-angiotensin-aldosterone system that operates to basically raise your blood pressure back up. Again, once you've had the blood filtered, it'll enter the Bowman space and then enter the PCT, which is this structure right here, and then it's going to loop all the way around and then enter this structure here, which is called the distal convoluted tubule. This one is the distal. Within the distal convoluted tubule, you have the macula densa, which is going to be communicating with the juxtaglomerular cells. It's going to be detecting things like your sodium and chloride concentrations, and if it notices they're a little bit off, it'll release something called a paracrine agent. You don't need to know that now, but for physio next semester, you'll talk paracrine and autocrine agents. They're a type of chemical messenger that are done in like a small location. So basically, these cells are communicating with their neighbors over here. So they'll release something that essentially tells the juxtaglomerular cells to release renin. And the macula densa is in your DCT. The juxtaglomerular cells are in the wall of your afferent arterial. Okay, who's next? Ooh, we definitely need to do the heart. is this an anterior view of the heart or posterior? Anterior view. Posterior view is going to look like this, where you'll get a really good view of this vessel right here, which is the inferior vena cava. Good. We can also see these vessels back here, which are the
Pulmonary.
say that again.
Pulmonary vein.
Pulmonary
Veins.
veins. Yeah, these ones are the pulmonary veins. These ones are the
Pulmonary.
pulmonary arteries. How do we know? Well, remember your pulmonary circuit's going to be one of the few circuits where the arteries actually are carrying deoxygenated blood, which our models always represent with the blue structures. Most of the time it's going to be a vein, with the exception of the pulmonary arteries and veins. So pulmonary veins are these ones, pulmonary arteries are these ones. What is this structure right here?
Superior
Superior
vena
vena
cava.
cava. What is this right here? The right
Atrium.
auricle, yeah, right auricle. What is this kind of groove on the center of, on the anterior portion of the heart, separating the two ventricles?
Interventricular.
Yep, or anterior intraventricular sulcus. I'll show you the septum in a second. What is this structure right here? What's the tip of the heart called?
Apex.
Apex. What would the flat portion be? Not flat, but yeah, the base. Let's open our heart up. Now again, all the auricle really does is just extend the volume of the right atrium, yeah. What is the valve that separates right atrium from right ventricle?
Tricuspid.
Tricuspid valve, also known as the. So if it's between an atrium and a ventricle, they also call them the atrioventricular valves. What are these strings called that connect to the capillary bodies?
Chordate tendineae.
Chordate tendineae. You've got this sort of band right here. Does anybody know what that's called? That's actually a papillary body. It's called the moderative band. Regardless, not commonly asked. What is like the kind of almost like, I think they said it looks like, I don't know, like raw meat.
Something carne.
Yeah, trabecular carne. And then what is this valve right here that separates the right ventricle and this vessel right here? Yeah, the pulmonary semilunar valve. Semilunar valves will not have chordate tendineae. What is this structure right here?
Pulmonary trunk.
Pulmonary trunk, and that's going to branch into
Pulmonary.
pulmonary arteries. What is this chamber right here?
Left atrium.
Left atrium. What about here?
Left
Left
ventricle.
ventricle. What are these called again? Chordate tendineae. What is the valve that separates left atrium and left ventricle?
Mitral.
Bicuspid or mitral or
Atrioventricular.
left atrioventricular valves. Now there is the
Atrial valve.
our aortic semilunar valve that separates the left ventricle and the aorta. So what is this first section of the aorta called? Ascending aorta. Then this is the aortic arch. What is this vessel? Brachiocephalic trunk. What is the middle one?
Left common carotid.
Left common carotid and
Left subclavian.
left subclavian. Very good. Okay, let's do that. Actually, the blood vessels next because
Oh my
we
God.
have those.
I don't know any of the blood vessels. Let's see. I know the femoral artery. It's like it.
I'm hoping we get one of the ones where it's like, it sounds like words.
Yeah, like, yeah.
I know I probably shouldn't bank on this,
Yeah, there's too many, like, veins is fine. Like, there's only like 10 veins we have to know, but, like, arteries is it's too much.
Too much.
I'm gonna memorize all of them just for there to be like one question.
Literally.
I just know like I'm gonna be so mad after the exam because it's gonna be like 10 times easier than I thought it was gonna be.
That's what I heard. I'd rather be more prepared.
So again, as you
Yeah.
guys are looking at
Yeah.
these, just remember, like, there's really not like this is exactly where it stops and this is where it begins. So look for anatomical regions that you know when you're trying to figure something out, particularly when you're doing like the upper extremity. So we said that this vessel right here, this one is the same as this one. What was that again? Brachiocephalic trunk. That's going to branch into this vessel here, which is the right common carotid, and then the right subclavian. So again, we don't have the bones, we don't have like the clavicle, but we know it's going to run underneath the clavicle here, so that's going to be your right subclavian. Now, what is the anatomical region for your armpit region?
Axillary.
Axillary. So then this portion would be the
Axillary.
right axillary artery. Next, we have this
Brachial.
brachial artery. What would this be?
Radial.
Right radial artery and the
Ulnar.
right ulnar artery. Then the one running down the middle. Remember what the name of that membrane was that connects your radius and ulna?
Intermembranous.
Interosseous membrane. So they just call that the interosseous artery. And then finally down here, you have the palmar arch. So the arm is pretty straightforward, I think. Unfortunately, we don't have a good vessel for like the blood or the vessels bringing blood to the brain. Like I don't think you can see this one. This is draining blood from the brain. Does anybody know what that's going to be called? Yeah, very good. The internal jugular vein. We know that because I can basically see that jugular foramen where it's exiting. This is the external jugular. I'm not going to ask you the branches of the external carotid artery. That is what basically this is showing you right here, where it branches off into those three sections. Let's do the abdominal aorta, because I think that's usually the trickiest section for students. Let's see what happened today.
What's this called again? Carina.
Okay, so once you get
C-A-R-I-N-A.
below the diaphragm, you enter into the abdominal aorta. If you're in that section of the descending aorta above the diaphragm, that's considered the thoracic aorta. So as you look at this, you have this first artery. It's kind of hard to see because it's branching into these three sections. Not there. The stomach is actually not shown in this model, but that's what this one would represent, which would be the gastric artery. Does anybody know what this organ is?
Spleen.
So that's gonna be the splenic artery, and then this is your liver. So it'll be the hepatic. What are those three arteries collectively called? The celiac trunk. Think of people with like celiac disease. I'm assuming it's related to that region. But that's called the celiac trunk, and it's going to branch into three unpaired arteries. Next, it'll be this one that kind of sticks out here. It's hard to kind of tell, but it'll look similar on the other model, though it's going to kind of be a little more separate from our renal vein right here. What is this artery? Good. Superior mesenteric artery. It's right there. Then you have your
Renal.
arteries. Now, on our other model, you won't see these two arteries. They are paired arteries called the gonadal arteries. On our other model, for whatever reason, they excluded them. I don't know if Dr. Gard requires you guys to know them for lab or for lecture either. Here's another unpaired artery. What is that? Inferior mesenteric artery. So superior mesenteric artery here, inferior mesenteric artery here. Then we branch into the common iliac arteries, internal iliac and external iliac. Now again, I could also ask you the veins, but for the most part, they're going to have the exact same names as the arteries. For instance, what do you think this vessel is right here? This one, external or internal?
External.
External, and this is the
Internal.
iliac veins. Obviously the inferior vena cava is going to have a different name, but what's this vessel here? Longest vein in your body.
Great Symphony.
Say that again.
Great Symphony.
Yeah, exactly. Great Symphony vein. Good.
Because like I was for everything.
Okay, how do you feel about blood vessels? Pretty good.
Could you just go for the arm veins? Because aren't those a little bit different?
Yeah, you do have the basilic and the cephalic, excuse me, yeah. I don't really know ask those, to be honest. Basilic. Not positive on this one. I believe cephalic's going to be closer to the head. I think that one is going to be up here, and then the basilic.
That one's like the one that runs along the ulnar part.
Like right here.
Yeah.
That would make sense. Actually, I don't know why necessarily that would be why it's called a basilic. But yeah, I haven't asked those, but I know other TAs have. So again, if you're not taking it with my class, like make sure you know those two as well. I wasn't required to know them, so I tend to be pretty consistent with what I was asked to know at the time, without knowing a couple of things that I find important. What have we not done? I feel like we covered.
What about the pancreatic model?
Oh yeah, let's look at that.
Yeah.
This is, I tend to, it is possible. We just lost the gallbladder.
Okay. Alright. I'm just gonna your turn.
Okay, that's fine.
Not really. Sorry, that was gross.
That's fine.
So here is, what is this structure right here? The duodenum. What is this organ right here? The pancreas. What is this duct right here? The pancreatic duct. And what is this?
Gallbladder.
What is this duct? What about this one? Yep, common hepatic duct, and we can see the two branches of the right and left hepatic ducts. And then this then makes this so cystic and the hepatic duct merge to become the common bile duct, yeah. That's pretty much the gist I'd ask. I guess maybe what is this right here? The spleen. How are we doing on time? Pretty good. We've only been going less than an hour. Let's do some histology stuff.
I don't know any of the histology. I don't know any of the
Is there
histology.
any histology you guys like want to talk about? Like off the top of your head? Very possible, yeah.
Did you feel like between the
Definitely. here's a seminiferous tubule. It's a little closer than I want. I think this image makes it pretty easy to see. These are seminiferous tubules. I wouldn't ask you guys basement membranes. Seminiferous tubules, again, I think I'd be more inclined to ask you it from this view, maybe a little bit closer. I just remember, like, it's kind of the same with the ovary where you start kind of from the periphery, and as you get more mature, you get closer to the center. So you've got, like, spermatocytes out here, and then notice as you're close to the center of this, you get the tubule, you have full spermatids. And how many, like, from one spermatocyte, how many spermatids do you end up with? 4. What about from one oocyte, how many viable ova do you end up with? 1. Now,
Are the seminiferous tubules like in the test as well?
yeah, they're in the testes,
Oh.
exactly. They connect basically to a set of tubules that'll eventually lead to the urethra to be ejaculated. Now remember your reproductive organs are mixed glands. If I were to ask you, like show you this histology and ask you, what is the endocrine secretion of this gland? What would
Testosterone.
I would ask you, what is the exocrine secretion of this gland?
Spermatozoa.
Spermatozoa. And this is just getting closer. We can see our nerve cells. Leydig cells, you'll notice, like you've got the tubules here. Leydig cells are kind of, they remind me of the like interstitial lamella, where you had like your concentric lamella surrounding but basically I'm thinking of compact bone.
Surrounding an osteon?
Thank you, osteon. So like in between the osteons, you have like the interstitial lamella. That's kind of what the Leydig cells look like to me, where they're like in between the different tubules. Let's look at.
Do we have any pictures like that close?
It looks pretty similar to that under our microscope, to be honest. I don't know if they'll be as well colored, but for the most part it should look very similar. Let's look at female ovary. Okay. How can you tell that this is a secondary follicle compared to a primary? What's something to look for?
The transition from the cuboidal to the
So they'll still be cuboidal. My understanding is they'll still be cuboidal, but you'll have multiple layers. In the primary follicle, notice how you have, they almost look like squamous cells to me, but it's going to be one layer. Whereas when you get into secondary follicles, notice how there's 2 layers of cells. That's something to look for. Primordial follicle, it just looks quite nebulous in my opinion. Right at the center, you have a primary oocyte, even in a secondary follicle. So there, notice this is a secondary follicle close up. There's 2 layers of cells. Okay, what then would this be? Tertiary follicle, or there's a couple names for it. I've heard tertiary follicle, antral follicle, or Graafian follicle. What would this kind of big sac of estrogen be called? Starts with an A. Antron. Here we have the corona radiata, and then we have our secondary oocyte. Again, same thing, just a different image. What is this called?
Antron.
The antron. There's our oocyte at the center. Then this layer of cells right here, they call that the zona pellucida. I haven't really asked that many times. Corona radiata is definitely a fair question. It's harder to get this to kind of be distinguished. More of the same.
Is it called the antrum even if it's not a tertiary follicle?
Most of the time I've really only seen them in like tertiary follicles. Like these are both different tertiary follicles. You can see this one just maybe a little bit more developed. But I remember there was a slide on the reproductive where it like kind of showed you on both sides of like an interim between secondary follicle and tertiary follicle. The antrum developing. But I don't know if it had a separate name at that point. Corpus luteum, corpus albicans. The difference is corpus luteum will be significantly larger, so I'd be unlikely to ask you them in isolation when you only see this. I don't ask the menstrual cycle, like secretory versus proliferative versus menstrual phases. And then we don't have a uterus histology slide, so I'd probably be unlikely to ask you guys about that under the microscopes.
What about the kidney?
Is that okay?
Will we have to know the leukopenny histology?
Isabel and I were looking at that histology the other day, and it's like the pictures are much easier to tell the difference between them. So I was thinking about maybe asking you difference in the tubules. Leukopenny, at least the thin limb would be easy because it's one of the few that it's like squamous cells. But for the most part, like you can see the fuzzier layers for the proximal convoluted tubule indicating the brush border, but it wasn't as easy as I thought it would be to see. So I was like, I don't know if I'll maybe ask this. I think glomerulus is a fair question. Like if I point to this, like you can clearly, like glomerulus is very easy to identify. And we see all those different tubules, glomerulus.
What's the space in between the glomerulus to like the balance and the capsules? Like the space of the lumen kind of differences?
Like here,
Yeah.
I mean, I think they just call that like the bone space or glomerular space. If I'm not mistaken. Yeah, so this is like what I was hoping. I guess I could show you this image. For instance, what do you think this vessel is right here?
Artery.
Not an artery. It's one of the tubules of the nephron. And I can see that fuzzy pink border, which is indicating it's a proximal. Because the proximal convoluted tubule, unlike distal, has the brush border made of microvilli. What does that do? Increases surface area for absorption. So that's why they call the PCT the mass absorber, because once your blood is filtered into the tubules, most of what's been filtered is going to be reabsorbed in that PCT. And it's partially facilitated by that brush border. The DCT does not have a brush border. Its reabsorption and secretion is going to be regulated by hormones like aldosterone, like ADH, all also AMP, atrial nitropeptide. Yeah, sometimes we'll see the distal convoluted tubule, they'll say it's smaller in diameter, but I mean, I don't really feel like it's smaller in this image, so again, I wouldn't ask you guys that. Again, for the thin limbs of the ascending, I think I even actually used a kidney slide all the way back on your guys' first lab exam when I asked about simple squamous. I think I literally used the thin limb of the Henley's loop, so kind of reconnecting that piece. Okay, let's look at, there's a lot of histology from the digestive system. Do you guys want to look at that? Okay, esophagus. Remember, you've got stratified squamous epithelium, and then it's going to be a thin layer of muscle right here. Again, esophagus can be hard to see; it's not as well stained. But that is all of the epithelial tissue. Again, I would ask you what's this outer layer of the esophageal walls. That's the adventitia. Yes, definitely. So that'll be in our respiratory system.
Well, with the submucosa and sublingual, those glands.
The salivary glands. Yeah, I mean, that was something I was not asked about, so I tried to like be consistent with, like, there are ways to identify the difference and we can look at them, but in the past I personally haven't asked about, like, is this the sublingual gland versus the mandibular versus the how can we tell this is the pseudostratified? How can we tell this is the trachea? I look for the hyaline cartilage, and then I also look for, you can see the pseudostratified columnar ciliated epithelium. It's not going to be super zoomed in the way that it was for your first exam, which I used the trachea slide for your first exam when I asked you about pseudostratified ciliated columnar. But look for those two in conjunction. That's a very good sign. You're looking at the trachea. Okay, stomach. Again, I typically don't ask, is this the cardiac stomach or the floor portion of the stomach via histology. There was one TA that did, but I don't think she's one of the TAs anymore. Look for glands, particularly in the caloric region. You can see mucous neck cells, the gastric pits. There's that model, so everything's labeled here. Now, small intestine again, we can see those finger-like projections, those villi. Be able to identify the duodenal glands or the glomerulus glands here, muscularis externa, and then serosa. So again, how can you tell this is the duodenum? Look for those glands, those Brenner's glands, the duodenal glands. Same image, just a different color. jejunum. Now the jejunum, unlike the other ones, they don't have like a specific gland that I know of. They will have very obvious villi. There's these intestinal crypts, but they won't have, like, notice their submucosa is pretty thin. And they're not going to have Peyer's patches or Brun's glands, to my knowledge. I always felt like the submucosa was particularly thin for the jejunum, but I could be wrong. Okay, under the ileum. Like those niggles. I think ileum's usually pretty easy to see because of the Peyer's patches. Okay, and then again, once you get into the large intestine, notice how the villi pretty much go away. The large intestine mainly functions in compaction and storage of feces. Now you can have more reabsorption of water, but for the most part, it's not going to be associated with like the level of absorption that your small intestine did. Same layers, but again, notice you don't have this super long villi now. One thing I didn't go over was ocular glands. Sublingual is going to be directly beneath the tongue here. Submandibular, think there's our mandibular bone, so that's going to be our submandibular gland. Carotid is not shown here. There's a carotid gland, carotid duct. Does anybody remember the gland that makes the most of your saliva? Submandibular. So I guess I didn't include slides on the pictures of the histology for the glands. So since I didn't, I'll stay consistent and not ask you to be able to identify like this is the submandibular versus sublingual. But again, if you don't, like for whatever reason you're sick and you're not able to take it with my class, just I could definitely look that over because another TA can ask me about that. Okay, let's look at our lung histology. In alveoli, what do alveoli do? What are they?
Ear sacs.
Say again?
They're ear sacs.
They're air sacs, and those are going to be made up of type 1 alveolar cells, and those are the ones responsible for the actual gas exchange. What about type 2 alveolar cells? What do those do?
What's it called? It starts with S, I think. They don't secrete. I don't know what it's called.
What do they produce that is necessary to keep your airways open?
Something that starts with S.
Surfactant.
Yeah.
Type 2 cells produce surfactant.
If I had
Those
a multiple
basically
choice here, I would
get in the way of like the interacting water molecules in your lungs. So again, if you've ever washed like metal dishes and when you try to pull them apart, they stick together. That's due to the interacting water molecules. Surfactant essentially gets in between those, so it prevents your lungs from sticking together after you've like taken a breath out.
And what did type 1 do?
Say again?
What did type 1 do?
Type 1 cells are the actual cells that are doing the gas exchange. So it might have been I used alveoli for simple squamous on your first exam. It was either that or the kidneys. I can't remember which one I did. I think I switched out for the classes too. But your like type 1 alveolar cells are simple squamous cells, which makes sense because they're super thin. They're perfect for gas exchange.
Can you tell the difference then between type 1 and type 2?
Most of the time, type 2, nobody's ever pointed it out to me. I'm sure there is a difference, but oh, here we go. Type 2 cell. So this big guy or nucleus, I guess it just looks a little bit more cuboidal, whereas here we can see that super thin squamous cell of an alveoli. That's cool. I'm actually never taking a good look at this.
might get another
I wouldn't
kitten.
ask you guys to know the difference, but you will need to know it for lecture, like what do type 1 cells do, what do type 2 cells do.
I put in an application, but I guess like the post went like viral and like a bunch of
Okay.
people applied.
Blood vessels, again,
But,
unlike
like,
our
I'm
ducts,
hoping
are going
that,
to be
like,
full of.
They'll pick
you'll
me
notice
because
there's
they know I'm
tissues
not
in
giving
the
it to
center.
kid. It's a girl.
As you're looking at like the
So that's not
respiratory
gonna be like a Christmas
bronchioles
present
as
for a little
blood
kid.
duct,
So
usually it's
I'm
just
like,
thicker tissue around the bronchiole, and then as you get into the ducts, it'll kind of thin out.
Well, I just put because I like asked for like my house, like
So here's terminal.
where I live and who lives in the house with me. So like I just put it to myself. I mean I'm a 21 girl. Like I'm not gonna buy it for a kid.
I know, because then it doesn't sound like you're like a responsible like college student, like
Yeah.
just getting a group kid or something.
Yeah, there wasn't like a description for it, so I think they have to email me, and they'll probably ask more questions. So they'll be like,
I hope you get it.
yeah,
Sorry, I'm trying to also follow. I'm listening.
fine.
Also, if you want, I can, because you have if you want, I can send you the document with all the pictures.
Okay,
Or like email it, whatever.
okay.
Which one is you?
For the histology of the lymph nodes, again, I would know the cortex versus medulla. A way to tell the difference is you can see the lymphatic nodules will be in your cortex. I think that's kind of the hardest question Dr. Yarda allows us to ask.
The lecture exam has so
Lymphatic
many
system.
chapters.
Wait, you said is the nodule that dark?
It's like 9 chapters.
Yeah,
or something.
you'll see.
Lighter area on the inside would be good.
Medulla, yeah, the medulla, it should be sterile, obviously, just no nodules
Really?
in the
Yeah.
cortex
She,
portion of the
she
lymph
gets
node.
like, she's like a perfect score in every exam.
That's crazy. I mean, I think I need to get like, I think I can get like damn near like a 40%.
And then finally, you guys will need to
When he
know
hurts it.
I'm excited for that.
arteries versus
Yeah.
veins, the various layers of the walls of these vessels. So tunica adventitia, tunica media, tunica intima, or tunica interna. Sometimes it's obvious, sometimes it's less so. But I could ask you, what is this vessel? What, and then have the arrow on like a specific portion. In which case, I'd ask you, like, what layer is this? So bear that in mind. Again, a lot of times we'll say, oh, well, the artery is easy to tell because it'll maintain its circular shape. Sometimes, not every time, so don't get thrown off if it's like not a perfect circle. For the artery, check in with like the thickness of the walls is usually a good cue, because the tunica media for an artery will always be the thickest of the 3 layers, versus for veins, it'll be that tunica adventitia out here. So even though this tunica media looks as thick as this one here, notice this layer is clearly thicker than that tunica adventitia. You can see a little bit of that internal or external elastic lamina between the tunica media and adventitia. So that's a cue. Look for that in case it's not, I don't know, it's not immediately obvious. Only all of our slides look exactly like this. This is very obviously a vein, very obviously an artery. Okay. I think that's pretty much it.
Would
Yeah.
would you ask like spleen histology or
I don't think we have a spleen slide, so I don't typically. I wouldn't ask it under the microscope, and
Did she say anything about
I generally won't ask it if it's not a possible microscope
I think she-
question
yeah.
to do.
and then what about
Just
like
in
the
case.
tonsils?
I don't think we have tonsil slides either. I would be able to recognize gross anatomy of the tonsils, so on like the half head models. But I wouldn't ask you the histology for the tonsils.
Okay.
Dragon question. Any other questions? How are we feeling? Pretty good. Usually goes a lot better for people. The multiple choice makes it a lot easier. But definitely know your rights and your lefts.
Yeah.
Sometimes I'll put a sneeze in them for the blood vessels, so just double check with yourself and don't listen to the point.
That's
Yeah. Any other questions? Okay, if you haven't turned your labs in, please do so now. And you know, if you have the rest of the time to work out, whatever it is. And you're also welcome to come by